Alrighty then. Let's take a look at chapter 10. Okay. So chapter 10 is photosynthesis. And of course, we're going to start with a picture of a tree, right? Because leaves are the portion of the tree that conduct all of the photosynthesis. And I particularly like this picture because it shows all of the different kinds of organisms that are photosynthetic. And I think for most of us, plants and algae pop to mind fairly quickly. Let's look at them larger. Okay, plants like in the forest, right? Algae living in the ocean primarily, but freshwater algae as well that would be living in ponds and streams. Many protists, which are known as unicellular eukaryotes. These are pictures of a euglena, and all of these green things, all this green area that you see in here, those are actually the chloroplasts inside the euglena. They have flagella, they swim around, so they're a protist, so they're kind of in between a plant and an animal, if you want to think of it like that. There are bacteria, particularly the cyanobacteria. These are the modern relatives of some of the first cells on Earth that were photosynthetic. They also fix nitrogen, kind of super bacteria. And your textbook includes the purple sulfur bacteria to remind us that you don't have to use green pigments, or you don't have to use chlorophyll in order for your photosynthesis. Some organisms use other pigments, and plants use other pigments as well, as we'll see in a second. Okay, so sort of going back to plants, because they're the most familiar to us, right? We call this panel sort of journey to the center of the leaf. Okay, so if this is a leaf on a plant and we could cut into that leaf and look at it in cross section. So this is the top of the leaf and this is the underside of the leaf. Microscopically, of course. These are the chloroplasts. They're lined up in two layers of what's called the mesophyll. This is the palisade mesophyll because it's lined up like a fence. And this is the spongy mesophyll because the chloroplasts are all in cells that are kind of floating around in there. Notice another important feature of the leaf is this vein. We saw these way back in chapter 1 when we looked at adhesion and cohesion. These veins are used for water to be transported from the roots of the plant all the way up to each leaf, as well as for transporting the glucose that the leaf cells are going to produce down to the rest of the plant. The gas exchange, carbon dioxide and oxygen exchange, that the plant cell is going to use as part of its reactants in order to conduct photosynthesis. Those gases are exchanged through what's called the stomata. These are little openings on the underside of leaves that open and close in response to temperature and how much water is available. And they let carbon dioxide in and oxygen out. Oxygen is going to be a byproduct of photosynthesis, and carbon dioxide is going to be a requirement. It's going to be pretty much the opposite chemical reaction to cellular respiration. And if we look inside one of these teeny tiny mesophyll cells, it's full of chloroplasts. You can see they're just all floating around in there. This is the picture of the chloroplasts that we looked at back in chapter 6. This has been colored in different colors to show us the actual stacks of thylakoids, which are the working membrane. This is going to be the important membrane portion of the chloroplast. Chloroplasts have 3 membranes, or a triple membrane, outer, inner, and the innermost membrane folded up into these discs. Each disc is called a thylakoid. The stack of discs is called a granum. And the fluid around the discs is called the stroma. Okay, so we did learn that back in chapter 6, and the reason, here's some bigger pictures. And the reason is because we were going to need, just like with the mitochondria, we need all of these spaces in order to complete photosynthesis. Okay, overall chemical reaction for photosynthesis is going to be carbon dioxide plus water plus light energy. And the products are going to be glucose, water, and oxygen. I think on your handout, I have that actually, let's stop this share for a minute. I think I have that on your handout actually written out, the complete reaction. Let's see if I can find it real quick in your textbook. I think, yeah, it's pretty generic, but I'll just write it out anyway. So 6 carbon dioxides, and we're going to need 6 of them because glucose has 6 carbons, plus 12 water molecules, because we're going to use some and we're going to regenerate some, plus light energy. And there's going to be multi-enzyme steps here. That this arrow should have multiple arrows inside of it. And it's going to give us glucose, plus, as I mentioned, some water is going to get regenerated, and we're also going to release some oxygen. 6 oxygens and probably 6 water. Yep. So just like we learned the overall chemical reaction for cellular respiration, we also need to know the overall chemical reaction for photosynthesis. Okay, and this would be the version that I'd be looking for. You can also find it in your book on page 180. Now, just like we talked about with cellular respiration, these are oxidation reduction reactions. So what is going to be happening is the carbon dioxide is going to be reduced. So remember, reduction, oxidation involves loss, right? Reduction involves gain. So the carbon dioxide is going to be gaining hydrogen atoms in this case to be converted into glucose, and the water is going to be oxidized or losing the hydrogen atoms to become oxygen, free oxygen, plus look, only half of those waters. Okay, so probably the hardest part about photosynthesis is the beginning part, because it's different. Photosynthesis comes to us in 2 major phases or steps. So just like when we talked about cellular respiration and we went through 4 steps, we're going to go through 2 major steps here, and both of them have some subcomponents, particularly the first one. So the 2 parts of photosynthesis, we refer to them as, the first part is called non-cyclic photophosphorylation. Okay. Non-cyclic, not a circle, meaning the electrons do not go back to the molecule they started at. There are some cells that actually engage in cyclic photophosphorylation. So this is an important distinction. Non-cyclic photo, light, phosphorylation, adding a phosphate to something. Okay. This step 1, this first portion of photosynthesis, sometimes they used to be called the light reactions, because this is the portion that involves the light. Okay. There are 2 subcomponents to this non-cyclic photophosphorylation, and we call them photosystem 2, Roman numeral 2, like that, and photosystem 1. And you might think, well, why didn't she put 1 first? Because in the cell, in the chloroplast, what we're going to see is that photosystem 2 physically is first. So we list them: photosystem 2, then photosystem 1. The second major part of photosynthesis is the reduction of carbon dioxide part. And that portion is called the Calvin cycle. And as the name implies, cycle, because it's a circle, okay, similar to the Krebs cycle or the citric acid cycle. This is going to be a circle that we'll talk about at the end. The reduction of the carbon dioxide is the part where we actually produce the glucose. This part, the non-cyclic photophosphorylation, photosystem 2 and photosystem 1, this is all about producing, the outcome of this is all about producing ATP and, I have the yellow sun, and another electron carrier, NADPH, because it has an extra phosphate. So it's NADH with an extra phosphate. I'll put that one on the square. These are molecules that can then be used for energy to produce the glucose. So we're going to look at how the cells and the chloroplasts produce the energy first, using light energy, changing light energy into chemical energy. So converting energy from one form to another, the laws of thermodynamics, in a long pathway, so to minimize the heat or the entropy. Right? So this portion is all about producing the ATP and the NADPH. Then the ATP and NADPH are going to be used by the cells to actually drive the Calvin cycle to produce the glucose. How the cells make the ATP is the same as we're going to see, almost the same as we saw with cellular respiration. It's going to involve electron transport chain and passing electrons and pumping protons. So that part of it's going to look, that part will look familiar. The actual converting of the light energy into the potential energy that's going to be chemical energy, that's the part that's different and new. Okay, that's what we need chlorophyll for. Okay, so I think I'm going to go back to these pictures. So that's what this part of your book is trying to show you. They're trying to show you, okay, there's this portion, what we would call the light reactions or the non-cyclic photophosphorylation. And you can see it's happening in the thylakoid membrane inside the chloroplast. And they're showing you that ATP and NADPH are formed from this part. This is also where the water is actually split and oxygen is going to be produced and then released through those stomatas. And then the second portion, the reducing the carbon dioxide portion, we have to bring the carbon dioxide in order to produce the glucose. There we go. Okay, so now the how-to. How do we do this first part? How do we take light energy and convert it into chemical energy, convert it into ATP and NADPH? So we need to first learn or remind ourselves a little bit about light, about all the energy that comes from the sun. We call this the electromagnetic spectrum. And so this includes everything: gamma rays, X-rays, UV light, shortest wavelength to longest wavelength. The shorter the wavelength, the more energy. The longer the wavelength, the less energy. Okay? These are the wavelengths of visible light. If we just take this little bit of the spectrum right here and we break it out into the colors of visible light, we can actually know and mark all the different wavelengths in nanometers of the different colors of visible light. And you're like, okay, why do we care? Well, we care because only some of these wavelengths are going to excite chlorophyll. And chlorophyll is the major molecule that is going to help with this energy conversion of light energy into chemical energy. So this is the electromagnetic spectrum, includes all the energy that we get from the sun. The visible light range here is what we're most interested in photosynthesis, and what we're going to see is not all of this. Plants can't use all of this visible light, they can only use some of it. And in actuality, they can use almost all of it except the green. That's why chloroplast and chlorophyll looks green, because it reflects the green light, but it absorbs the energy from all of the other colors or all of the other wavelengths. So I'll say that a couple more times because it seems counterintuitive, right? We are seeing the green because that's what's being reflected or not absorbed by the cells. Most of the other colors are being absorbed, some by chlorophyll, of which there's two forms, A and B, and some by actually other pigments in leaves that we don't talk about as much as chlorophyll. Chlorophyll is the major one. So here comes the light energy from the sun. It's entering into the chloroplast, and it's going to be, you can see all the different colors here, right? The reflected, they're trying to show you the green is reflected, most of the other colors are absorbed. The absorption spectra for most plant cells, for most organisms that use chlorophyll A and B, this is the spectrum of what colors are absorbed. And you can see clearly here, chlorophyll A is this blue-green peak, and you can see that it does most of its absorbing of energy here in like the purple-blue area. And then there's another little blip here out at sort of orange-red. Chlorophyll B, the peak of chlorophyll B is offset a little bit more toward the blue and a little more toward yellow-orange. The carotenoids, these are other pigments, and you can see they also mostly absorb here in the purple to blue. They're reflecting green. They're not using green. They're sending those wavelengths back out, and that's what we see. That's why we see plants looking green. All right, so now let's take a look actually at what chlorophyll itself looks like. Okay, so we have two forms of chlorophyll in plants, chlorophyll A and chlorophyll B, and they look just like this. This is their chemical structure. This part might look kind of familiar to you, right? It kind of looks like fatty acids, right? A hydrocarbon tail. And this tail is going to allow the chlorophyll molecule literally to sit in the phospholipid bilayer, and the head portion that's highlighted in green here, it's also known as the porphyrin ring. This can rotate and sort of move around a little bit as well so that it can capture the maximum amount of light that comes in from the sun, the maximum amount of photons coming in from the sun. The difference between chlorophyll A and chlorophyll B, you can see here, is very small. Chlorophyll A, this is a methyl group. Chlorophyll B, less hydrogens, and we have an oxygen added on there. Okay, now I wanted to just actually physically show you this for a minute. So I'm going to draw a bit of a membrane here and put some chlorophyll in it. So we have our regular old phospholipid bilayer, right? But this being the thylakoid membrane. So that would be inside a chloroplast, right? So sort of like I drew the other day with the mitochondria, I would draw the thylakoid membranes like this in a stack, right? That stack's called a granum. The outer membrane, the inner membrane of the chloroplast, and then the innermost membrane. So this, what I'm drawing here, is just a little tiny portion of one thylakoid membrane in that stack of a granum. So in this membrane then, we're going to have what is known as a photosystem. And a photosystem is a set that also kind of looks like a tooth, and I'm making it purple on purpose because it's a bunch of proteins, and in this photosystem, embedded in this photosystem, are multiple copies of chlorophyll, right? So I always usually use my arm to represent chlorophyll, right? Because it sits in the membrane. This would be the hydrocarbon part, and this head portion is that flat porphyrin ring that can kind of rotate around a little bit to catch maximum light energy from the light rays coming in from the sun. Now, in this photosystem, there are multiple chlorophylls, and there's, we call this one the special chlorophylls, that when they get hit by light rays from the sun, here comes light energy from the sun, so I'm making it yellow on purpose. It's striking the chlorophylls, and these chlorophyll molecules then get excited, and their electrons, what we mean by that is their electrons start to move, and they get so much energy from the sun, from the photons, they can actually transfer that energy to their neighboring chlorophyll molecules. When that energy transfer, those photons, when they hit these two here, these special chlorophylls, they get so excited that 2 electrons are going to get sent to a further away energy shell than normal, and they're going to then be removed from chlorophyll, and they're going to be passed down an electron transport chain to some proteins that are here in the photosystem ready to catch those electrons. Okay, before we go and before I show you the rest of the photosystem, so this would be photosystem 2 here. I'm going to show you the pictures from the book to give you, again, a little bit of a mental rest and time to catch up to sort of where we are. Okay, so I'm going to go back to sharing the pictures from the textbook. So again, this is our chlorophyll molecule, and when light rays from the sun come and strike, when photons strike this porphyrin ring, some of the electrons that are normally associated with some of these chemical bonds, they get so excited that they can stimulate other chlorophylls next to them, and those special chlorophylls then are going to throw off 2 electrons. This is probably my favorite picture of this chapter. Oh, they changed it. I don't like that. All right, so this is the cartoon version of the photosystem 2, which looks just like my picture on the board, surely, right? With the photons coming in from the sun, the green dots being the chlorophylls, and you can see they're transferring the energy to the special pair of chlorophyll here in the middle, and see how they have an E for electron being sent up and out. Those are going to get, those electrons, that pair of electrons is going to get captured by a protein molecule sitting in an electron transport chain in the thylakoid membrane. This picture has all of the purple protein subunits that are all around the green chlorophyll. This is what it really looks like in 3-dimensional space, which I think is kind of cool. Right, you can see all the alpha helix and the beta pleated sheets, and it's a huge protein complex, this photosystem, right? So a photosystem technically is many molecules of chlorophyll with proteins embedded in a phospholipid bilayer of the thylakoid membrane. Okay, now what? Okay, next part. I'm going to skip all these. I think this, although it's the most complex of the pictures, is probably the easiest to look at all at once, especially if we remember what we were looking at last time we met, which was a couple days ago, when we were looking at this cellular respiration, the membrane of the mitochondria. It's very similar. See how this is a phospholipid bilayer? We have purple proteins in the membrane. Notice it's a much shorter electron transport chain, right? So we've got photosystem 2, this molecule PQ, another one of these molecules that's actually several molecules, but we're just drawing it as a large one in this little peripheral protein here. And notice that the electrons from photosystem 1, from the chlorophylls of photosystem 1, they're going to get excited, they're going to get passed. They're going to end up at photosystem 2. At the same time, electrons from photosystem 2 are going to get passed. They're going to end up at photosystem 1. At the same time that photosystem 2 is getting stimulated by light, so is photosystem 1 getting stimulated by light energy. And notice it's slightly different shape. Looks like it has more chlorophyll in it. But it also has a special pair of chlorophyll molecules that are going to send up a pair of electrons. They're only going to an even shorter electron transport chain here. And those electrons are eventually going to be used to reduce our friend NADP. To reduce it, because it's gaining hydrogen atoms, into NADPH. Notice the enzyme here, this last protein on the electron transport chain, is called NADP reductase, ASE, reduct, because it reduces NADP into NADPH. Just like the electron transport chains we talked about in cellular respiration, while the electrons are being passed, protons are being pumped from one side of the membrane to the other. Those protons are used as fuel. Oh, look, here's ATP synthase again, to produce ATP. It's just not sitting physically at the very end of this second electron transport chain from photosystem 1. It's just sitting a little bit of a distance, almost on the other side of the thylakoid space here from the electron transport chain. And then real briefly, and I'm going to redraw my cartoon version on my board of this picture, okay? Real briefly, notice that these two molecules, the NADPH and the ATP, that we produce from non-cyclic photophosphorylation, see how they're headed to the Calvin cycle. The Calvin cycle is going to take place in the stroma. Right here, the stroma. The photosystems, photosystem 2 and photosystem 1, take place in the thylakoid membrane. So the non-cyclic photophosphorylation takes place in the thylakoid membrane. Calvin cycle takes place in the stroma. I wanted to be sure to say that so that I didn't forget later. All right, so now you're going to have to look at my poor picture diagram of this, my recreation. How well I can do here. All right, so this is my photosystem 1. I should have kept it further off to that side, but nonetheless, I've got a little PQ here. And then very close to that, I'm going to draw this other one. It looks like that. And then there's one underneath here. That's PC. And then very close to that, I'm going to get my photosystem 1, which like that. Out of room. All right, my apologies for this line. I'm trying to draw this phospholipid bilayer here. And I'm going to put my green chloroplasts or chlorophylls in photosystem 1 over here. With a special pair in the middle. Put some light energy on here, bouncing electrons around, and we're going to throw up 2 electrons that are going to get passed. These electrons, right, left the chlorophyll, they get passed to PQ, and they're going to get passed to this guy. They're going to get passed on, and then they're going to end up here at photosystem 1. Just digest that for a second. So the original electrons from photosystem 2, from chlorophyll, photosystem 2, they got so excited they get passed, and they end up here at photosystem 1 because they are the replacement electrons for the 2 that photosystem 1 gave away to NADPH. NADPH That's the recipient of those electrons. That's what reduces, that's what this molecule is doing, that last purple blob is doing. It's reducing, it's taking the 2 electrons, adding them, plus the protons, adding them to NADP, so that it becomes NADPH. Okay, now, if we think about this for a minute, if I take 2 electrons away from photosystem 2, and they're being used to replace the electrons from photosystem 1 that ended up to make our NADPH, wonderful. Then I can only do this one time. I have to have a way to keep feeding electrons to replace the 2 that get sent away from the excitement from photosystem 2 that end up at photosystem 1 that eventually end up at NADPH. The replacement electrons for the whole system, but particularly for photosystem 2, they're going to come from water molecules. Water is going to be split. That's where the oxygen comes from. That's going to be sent out as waste. But the hydrogen atoms, the protons plus their electrons, 2 of them, those are going to replace, they're the replacement electrons for photosystem 2. So that when light strikes photosystem 2 and photosystem 1, this can all run again and we can make more NADPH. Okay, so technically, if you want to think about where do the replacement electrons for photosystem 1 come from, they come from photosystem 2. Where does photosystem 2 get its replacement electrons? From splitting water. There's an enzyme for that. And it happens right here in this beginning of this photosystem. Now, the electrons from the splitting of the water are headed to replace the electrons from the chlorophyll, but the protons aren't, the positive charges aren't. Those positive charges, that's what's going to be used, as well as, I think your book shows, there's also some protons pumped from here. And they kind of show it going around PQ like this. So protons from splitting the water and protons get pumped across the membrane as well. That's what's going to fuel ATP synthase, which, right, in our picture of the thylakoid in your book, right, this kind of, the ATP synthase is sitting down here. Draw this with the connection with the round part here, and then that arm, right? And ATP synthase is going to be using the protons, right, to turn to produce ATP. I have a yellow sun here. Let's look at that picture again now, at the picture from your book. It's a little more complete than mine. Right, so light energy striking photosystem 2, exciting electrons. Those electrons get so excited in the special chlorophylls, they jump to these proteins. They get passed, and they serve as replacement electrons for photosystem 1. Why? Because photosystem 1 also, the chlorophyll was excited, so excited that it threw electrons to a short electron transport chain. And those electrons are gone. They're here. They have reduced NADP into NADPH. So if ever the plant cell wants this to happen again, we need to keep feeding replacement electrons to photosystem 2, so that photosystem 2 can keep feeding electrons to photosystem 1, so photosystem 1 can keep adding them to NADP to cause it to become NADPH. So this is, in essence, this is why plants need light and water. I'm saying that they need light and water to produce ATP, to produce energy. They're actually producing 2 forms of energy, NADPH and ATP, and then they're going to produce glucose, which other parts of the plant can then use in their mitochondria to make other kinds of ATP for the plant to make proteins and lipids and all the other molecules that it needs to make. When the water gets split, the electrons go to the chlorophyll molecules, right? The oxygen is released, and the hydrogens are going to be used to make a hydrogen gradient to pump through ATP synthase so we can make ATP. There aren't numbers associated with how many are produced because this system is in every single thylakoid. So if we go back to this picture, right? This system, there's multiple sets of photosystem 2 and photosystem 1 in each thylakoid, and you can see, right, there's many thylakoid membranes and there's many stacks of thylakoid membranes in these grana inside a single chloroplast. And if we think back to the journey to the center of the leaf, each mesophyll cell, this is magnified and multiplied. And then there are multiple mesophyll cells in each leaf. And then clearly, if I'm a tree, there's going to be, I'm going to need millions of leaves in order to produce enough ATP to make enough glucose to be a tree. Okay, now that ends the energy producing part. The second part of photosynthesis, remember I said, was the Calvin cycle. And the Calvin cycle, here's a picture of the Calvin cycle for you. The ATP and the NADPH that we produced from photosystem 2 and photosystem 1, so from non-cyclic photophosphorylation, that is going to be consumed in the Calvin cycle. Now, for the Calvin cycle, what do you need to know? Okay, you need to know that just like when we did the citric acid cycle, in order to produce a single glucose molecule, the Calvin cycle needs to be turned twice. So I would put up, and we'll put up the Calvin cycle. And similarly to how we did with the citric acid cycle, I would put a little table over here on the right for the Calvin cycle, and I would write 1 turn, 2 turns. So in most textbooks, you'll see they're going to put 3 carbon dioxide molecules into the start of the Calvin cycle. The molecule that they join, I just want to be sure to spell it correctly. The molecule that they join has a very long name, and you need to be able to recognize it, meaning you need to be able to see it in a multiple choice question and pick it out. Okay, that molecule is called ribulose bisphosphate. I'm going to draw it here. Ribulose because it's based on ribose, right? Bis because there's 2, and phosphate because it has phosphates on it. It is abbreviated. You will see it abbreviated when you go through the exercises, the homework, RUBP, like that. People abbreviate it. This molecule has 5 carbons. Okay. It is joined by these incoming 3 carbon dioxides to form the first molecule on the Calvin cycle. And that first molecule is a super, super short-lived intermediate. You don't need to know its name. And again, just like with the citric acid cycle, I'm going to draw some random arrows going around the Calvin cycle. And the things that we need to pay attention to with the Calvin cycle are the following. Here, right at this almost very first step, ATP needs to be inputted. So energy is required to turn the Calvin cycle. So the opposite of what we had with the citric acid cycle, where we had energy molecules coming out, we're going to be shoving energy molecules in, potential energy in. 6 ATP need to go into the Calvin cycle right here, right at the very beginning. And your book will put 6 ADP for adenosine diphosphates come out. The very next step requires the input of 6 NADPH, 6. Now, rather than energy coming out at the bottom, what comes out at the bottom is half of a glucose molecule. So I'm going to put 1/2 glucose. That 1/2 glucose has a name, and you have to know its name. Its name is glyceraldehyde 3-phosphate. This time the 3, not because there's 3 of them. See how it's 3 with an attachment to the phosphate? That's because this is telling you which carbon the phosphate is attached to. Half of the glucose. Glucose has 6 carbons, so glyceraldehyde 3-phosphate has 3 carbons. So this is a 3-carbon molecule. In order to get the Calvin cycle to turn again, over here on this side, right before the formation of the ribulose bisphosphate, another 3 ATP are inputted, need to be put into the Calvin cycle. So for a single turn of the Calvin cycle, we would require 6, if I put ATP here in the middle, NADPH here, carbon dioxide, and then I'm going to leave the, you can abbreviate glyceraldehyde 3-phosphate as G3P. That's the same as that. For one turn of the Calvin cycle to make half of the glucose, or 1 glyceraldehyde 3-phosphate, I'm going to need a total of these 6 ATP plus 3 more. I'm going to need 9 ATP, 6 NADPH, and 3 carbon dioxide molecules. In order to produce a full glucose, I'm going to need twice that amount. I'm going to need 18 ATP, 12 NADPH, 6 carbon dioxides. That's going to give me 2 G3Ps, which the plant cell will take away and make glucose. C6H12O6. And I would remind you that this portion, the Calvin cycle, takes place in the stroma. Okay. And that is where we are going to end chapter 10. Okay. So in the reading, I left concepts 10.4 and 10.5 in the reading, but I will not be asking you questions in particular about section 10.5 is just a review of photosynthesis, but 10.4, I will be asking you about questions about that at this time. Okay, I'm going to stop. Okay, so I'm going to stop. Just look at this picture, which is a little neater than the one that I had drawn, right? One turn of the Calvin cycle. We need 3 carbon dioxides, 9 ATP, 6 NADPH, and we're going to produce half of the glucose, which is 1 glyceraldehyde 3-phosphate. If I turn the Calvin cycle twice, I'm going to need double of all those things, and that is what is going to allow me to actually produce glucose. Okay, I am going to stop the recording.